Very good afternoon to everyone, and thank you for joining us today. After a very fulfilling and enriching day yesterday, we have come to the second day of Knowledge Summit. Before we go any forward, let's take a few moments to recap what exactly happened yesterday. We started our day with a plenary discussion where our guest speakers talked about bilateral cooperation between India and France in science and technology. We then had two panel discussions on open science and health and society. These two sessions brought together panelists from India and France who deliberated on the importance of open science practices to access and share research data and the importance of communication in raising health awareness in society. We also had a keynote lecture by Professor Bhattacharya on strengthening the mathematical foundations for enabling field deployable AI models. The second day of the summit is dedicated to another conference on Tara Oceans and scientific workshops on themes like One Health, marine sciences, artificial intelligence, sustainable energy, and green chemistry, biotechnology, and natural resources. To deliver the talk on Tara Oceans, we have with us today Dr. Chris Bowler. He's the research director at French National Center for Scientific Research. He is also the director of the Plant and Algae Genomics Lab of École normale supérieure and one of the scientific coordinators of the Tara Ocean project aimed at exploring the biodiversity and evolution of plankton in the world's ocean. His main research interest is to understand the response of plants to environmental signals thanks to genomics. Before I give the floor to Dr. Chris Bowler, I would like to let our audience know that we will be taking up a few questions towards the end of this talk. So if you have any questions, you can leave those in Ask Your Questions tab that you see on the right-hand side of your screen. Without any further delay, I now give the floor to Dr. Bowler. Over to you, sir.
Thank you, Rajni. And I hope you can hear me all okay. Very good. So it's a pleasure to be with you here today. I'm very sorry that I'm not in Bangalore with you. I used to be a regular visitor to Bangalore before COVID. I have collaborations with IIS, with NCBS, also with the company Avestajan, which probably many of you have heard about in Bangalore. So I'm going to talk to you today about Tara Oceans from distant in Europe, but I hope we can be together for this next 30 minutes. My talk will touch several aspects that will be discussed later on today concerning ocean health, human health, the links between ecosystem health and human well-being, some aspects of marine biotech and importance of exploring marine ecosystems, and also use of computational approaches like artificial intelligence, which are really important for us to explore the very, very large data sets that have been generated through this project, Tara Oceans. So I will give you a general overview of the project and some of the key findings that have been made from the project. So just as background, Tara Oceans has now been a project since more than 10 years now. It began with a 4-year expedition quite unusual as a project, on a research sailboat called Tara, which also passed by India during this 4-year expedition through Mumbai. So our goal during this 4 years of expedition was to explore marine planktonic ecosystems and to learn more about their organization and their sensitivity to climate change and other anthropogenic impacts. We did this through the collection of plankton samples, which were then analyzed back in the labs using genomics technologies and using microscopic imaging, high throughput technologies. All in all, we worked with about 35,000 samples that were collected during this 4 years of expedition. In addition to the scientific aspects, we also did a lot of activities around popularization of science, explaining science to general public, and also towards young children. And we also made various attempts to influence policy with respect to governance of ocean as well, which is very important for the science to translate into governance activities for better management of our planet. So Tara Oceans was initiated by Eric Carcenti, who managed the project for the first 10 years, and I have recently just taken over as scientific director of the project. So in these last 10 years or so, we have done a lot of work. We've generated many publications. We have 130 publications now describing some of the results of the project. Many of these papers are in very high profile journals. I don't want to go through much of the information from these publications. But in a nutshell, the kinds of things that we have generated are sort of summarized here very, very. I think Tara Oceans is the real significance of the project is that it is kind of the first end-to-end description of a continuous global ecosystem. At a planetary level, we were exploring this continuous ecosystem, looking at all of the organisms present ecosystem, from the very smallest viruses up to the largest kind of zooplankton, small multicellular organisms. Okay, and it was the first time that anybody has taken an undertaking such as this. So through our studies, we have described the microbiome of the ocean, which from prokaryotic side, the microbiome consists of about 35,000 taxa, containing around 40 million genes. The eukaryotic microbiome, consisting of unicellular, single-celled eukaryotes, consists of about 130,000 taxa, containing about 116 million genes. This is where we're at today in terms of sequencing this, the information from the ocean. And in terms of the viruses, we have described up to now about 200,000 different kinds of viruses coming out of the ocean. So all of these resources have really revealed the incredible discovery component of the project, because most of what we find is actually new. Most of what we find, the genes, the organisms, the viruses, had never been described before. So there's a huge amount of new information to be discovered and to be analyzed in subsequent years and decades. So importantly, all of these resources that we have generated are also now publicly available to anybody who wants to use this information. So this represents the largest ever DNA sequencing effort for ocean sciences. There's more than 160 million genes which can be publicly downloaded and studied, and we have also many millions of images of organisms in different kinds of microscopes that also can be studied and explored for information. And the kinds of discoveries that have been made by other people, not by the Tara Oceans group of scientists, some of the examples of amazing discoveries that have been made, I just want to show you in a couple of slides. One of them is discovery potentially of the precursor of all mitochondria. So we know that all eukaryotic cells contain mitochondria. They are the energy generating machines of all cells, all eukaryotic cells. We believe that they are derived from an endosymbiosis of a prokaryote, bacteria. But there's been debate about what sort of bacteria that was at the origin of all these mitochondria. In studies from Tess Ettema's group in Sweden, he found some new kinds of prokaryotes which are potentially closer to the origins of mitochondria than any of the alpha proteobacteria that had been discovered before. So these were organisms that he found in the middle of the Pacific Ocean, in the middle of Atlantic Ocean in some of our samples. And this gives you then an idea of the incredible discoveries that can be found by exploring our data. And another example of very surprising finding is discovery of new kinds of viruses, in particular a new kind of bacteriophage, which infect bacteria, which have very large genome sizes. These are new kinds of bacteriophages never before discovered. They were discovered in particular by Jim Field in the US. And then subsequently, Jennifer Doudna, who you will all recognize as being one of the winners of the Nobel Prize for CRISPR-Cas genome editing, discovered that these giant phages also contain CRISPR-Cas genome editing systems, which can be potentially used in future applications of genome editing. So this gives, again, a nice example of the kinds of unexpected discoveries which can come from exploration of these data sets. Coming back to the work that we have been interested in particular, let's talk for a second about photosynthesis. I'm sorry about my noise on the computer. So let's talk for a second about photosynthesis in the ocean. We realize from satellite imaging of our planet that the oceans contribute about 50% of the photosynthesis on the planet. The other 50% is from trees and plants on land, of course. So this 50% of photosynthesis, which is very interesting very important for producing food, the biomass that all of the higher organisms feed on, is very important to learn more about. We have some examples of the kinds of organisms that are photosynthetic in the ocean. We have information about different kinds of photosynthetic organisms, going from photosynthetic bacteria like Prococcus, like Synechococcus, nitrogen-fixing cyanobacteria like Phytodesmium, and different kinds of single-celled eukaryotes which perform photosynthesis, which are the equivalent of the plants, the trees on land. So some of these are diatoms, you can see in bottom left. Some of them are green algae, you can see in top middle. Some are dinoflagellates and so on. Throughout our oceans data, we could get a global survey of the importance of each kinds of photosynthetic organism at global scale and their distribution. So here we see a general maps of distribution of different kinds of photosynthetic organisms using the same marker gene to catch all of them. It's kind of challenge because some of these organisms are prokaryotes and some of them are eukaryotes. So it's difficult to find one single marker gene which will capture all of these organisms at the same time. But here we've done this using PSBO gene. And here we can see distributions of these different groups. So this is Synechococcus, Prochlorococcus, the prokaryotic groups. We see they're principally dominant in the lower latitudes, is where they're most abundant, whereas the larger organisms, eukaryotic organisms like diatoms, tend to be more prevalent in higher latitudes like in Arctic, as you see here. We can also take this analysis one step further and see how these different kinds of photosynthetic organisms separate out by size. And so we see here a size fractionation going from the smallest size fractions, mainly representing the prokaryotic organisms at the top, all the way down to the larger eukaryotic organisms in the bottom. And we can see like that the cyanobacteria are principally in the smaller size fractions, principally in the latitudes, the warmer regions. Whereas as we get higher size fractions, we see the larger eukaryotes, in particular the diatoms and the haptophytes coming up, in particular at the high latitude regions. So this gives you an idea of the relative importance of each of these groups as we look in different regions of the ocean. What was also very significant from our studies, and which we hadn't really realized before, is the importance of symbiotic associations of photosynthetic organisms with other non-photosynthetic organisms, with photosynthetic organisms. Symbioses, for example, between dinoflagellates and groups like foraminifera, groups like radiolarians, which have these beautiful cell wall exoskeleton structures, which have these symbiotic algae associated with them. We didn't really realize the importance of these symbiotic associations before we started exploring the Tara Oceans data sets. So this gives us an idea of the global sort of contributions of different kinds of photosynthetic organisms to carbon fixation in the ocean. But of course, photosynthesis is only one side of the carbon pump in the ocean. What is also very important is the export of carbon from the top layers of the ocean down to the bottom of the ocean. This is known as carbon export. It's a very slow process, but it's very important, ultimately generating all of the oil and the gas, fossil fuels that we are so actively burning, consuming today. So we have some very general ideas about how this can work. Carbon being fixed by photosynthetic surface layer of the ocean, those photosynthetic organisms being eaten by zooplankton, and then through decomposition processes, these carbon is then seen to the bottom of the ocean. But we thought with the Tara Oceans data set, we could try to get some better idea of which kinds of plankton organisms most strongly associated with carbon export activity. We could do that because we generated throughout the Tara Oceans transit, we generated data about carbon flux in the ocean, going from surface down to 500 meters in the water column. So we have quite precise data how much particulate material is in the water column at each of these different locations as Tara went around the world. So we have this data on the one side. On the other side, we have all of this data from the plankton communities, the different kinds of prokaryotes, the different viruses, the different eukaryotes, and how they maybe are interacting with each other in these interaction networks. So we thought we could put all of this data together and do a very large compute exercise, go from this sort of more general picture of planktonic communities and how they may mediate carbon export in the ocean to a more network-based vision of which organisms are involved and how important are those organisms in the whole community of organisms that is important for carbon export activity. So this allows us to identify, hubs which would represent taxa, which are particularly important for maintaining this network of organisms involved with carbon export. What we found from this study was, on the one hand, we found the kind of organisms that we expect in terms of radiolarians, dinoflagellates, but we also found organisms we didn't expect, like the cyanobacteria Synechococcus, tiny little organisms, and even smaller synechophages, the viruses which infect Synechococcus, also appear to be very important in being responsible for strong carbon export activity in the ecosystem. So this was a surprise in sort of helping us to link carbon fixation, photosynthesis with carbon export in the deep ocean, and the importance of different kinds of organisms. We can move from carbon fixation to nitrogen fixation, which also is really important. Just like on land, you all know there's a wide range of leguminous plants on land that fix nitrogen, generating different kinds of food for us to eat. They fix nitrogen through symbiotic association with rhizobium, bacteria in the soil. In the ocean, we also have nitrogen fixation. It is at least as important in activity in terms of generation of fixed nitrogen as we see on land. But the organisms that are doing this through symbiosis mainly are microscopic, so they're difficult to kind of capture and difficult to get general idea of how important each of these different kinds of organisms are. So again, we can do that with our Tara Oceans data set. We can use microscopic imaging data like this example here of a diatom with nitrogen fixing bacteria inside each of the cells, shown in green. And we can compare this with genomics information, and then we can generate maps of the relative contribution of each of these different kinds of symbiotic association in terms of the general contribution to nitrogen fixation. And so here you see different kinds of nitrogen fixing organisms. You can see very generally that their relative contributions in different regions. So, for example, the Trichodesmium cyanobacteria are especially important in Indian Ocean, as are also these diatom diisotroph associations, very important in Indian Ocean, whereas other kinds of associations are more prevalent in Pacific Ocean, for example, as you see here on the left. So this gives us an idea of the relative importance of different kinds of symbioses for nitrogen fixation in the ocean. So through these examples, you see what we can learn about nitrogen fixation, about carbon fixation in the ocean. The final topic that I want to cover in our presentation today is what we can learn about climate change in the ocean, climate change on marine biota in the ocean. I'm going to give you 3 examples, if I may, of what we have done with Tara Oceans with respect to climate change. So the first example concerns evidence we have for effects of ocean acidification. So we all know that the oceans are acidifying. This is just because of more CO2 entering the ocean, which acidifies the ocean. But it's quite difficult to get an idea of what effects acidification is having on the organisms, because we don't really have long-term time series to compare how the ocean was before Industrial Revolution with how the ocean is today. But we have one interesting example that has been used by colleagues in Natural History Museum in London, where they took our samples collected on Tara, and they compared them with samples collected on HMS Challenger expedition about 150 years ago. So they took samples from the same locations in the Pacific. They looked specifically at calcification of foraminifera in these samples. So foraminifera, they produce calcified exoskeletons, which you can see in these images here. And we know that calcification is sensitive to acidification. And so what these researchers did is they compared the foraminifera collected by HMS Challenger, you know, like 150 years ago, with foraminifera collected by Tara a few years ago. And here we can see a comparison of the thickness of the calcified exoskeletons of these organisms between the Challenger samples and the Tara samples. And you can see very clearly that these calcified cell walls are much thinner than they were in the past. So this is one example, I think an important example of where we have evidence of the impacts of ocean acidification on marine organisms over sort of these long times. A second example of that I want to give is how ocean plankton may change in the future as consequence of climate change. So thanks to the Tara Ocean's collections of samples, we now can generate a global map of the distributions and diversity of different kinds of planktonic organisms, for example, bacteria, parasitic protists, or copepods, things like that. And here we can see these global maps where we're looking at diversity gradients. And so what we see in general here is that we have higher diversity in tropical regions. As we go towards the higher latitudes, Arctic, Antarctic, the diversity goes down. This is latitudinal gradient of diversity, which is well described on land, which we also see in the ocean through these studies. So with these global maps, we can take this information, we can then project into the future ocean by using the simulations of IPCC climate models, in which the ocean changes are predicted end of 21st century in terms of changes in sea surface temperature, in terms of changes in production in the ocean. And so we can project what changes in diversity may occur of these different organisms as we move forward in the 21st century. And in these plots here, we see anomalies of projected anomalies of diversity of these different organisms by end of 21st century. So if we look to copepods, for example, this group of plankton, we can see general decreases in predicted around the lower latitudes, tropical regions. Those increases of diversity are predicted towards the high latitudes. In the bottom here, we've plotted this out by latitude, and we've compared this with other important processes in the ocean, like carbon export, like fisheries, fishing, and like density of marine protected areas global scale. Maybe you can see this a bit better in these global maps here, where you can see carbon export activity plotted over the ocean, fisheries, fish catch, and we can see the density of marine protected areas as well as the final plot. You can see that the projected anomalies in diversity predict up to end of the 21st century as a consequence of climate change are very much likely to affect carbon export, our fisheries, fishing industry, and also, our efforts to conserve marine ecosystems through the designation of marine protected areas. So this is one example of the kinds of things we can do with it. And then finally, let me give you an example of what we have learned about Arctic plankton through the studies of samples from Arctic. So we have collected samples from Arctic regions in 2013, from Russian side, from Canadian side. And with this data, we again did the same kinds of exercises in generating interactome maps of how organisms interact with each other within these plankton communities to see, who is interacting with who, what kinds of symbiotic associations are happening, what other kinds of associations may happen between the organisms. And what you see on the right here is sort of this giant hairball with each dot representing a different organism and the lines between them representing an interaction. Here we can see the whole plankton interactome at global scale taken from all of the samples from Tara. Now, if we plot onto this, the temperature at which those are generated, we see very clearly that we have two kinds of communities, okay? The community that in Arctic is this community here, which is really associated with low temperatures. So what you can see here is that there is really a different community of organisms present in Arctic ecosystems than there is in these more temperate systems, low latitudes. So it's really a different kind of ecosystem that is present in. What we then did in this study was to sort of simulate environmental change on this community by attacking it. So in this case, we could attack it by changing salinity, attack it by changing temperature, attack it by changing different parameters, and see sort of if the network disintegrates, look at its sensitivity to change. What we see in particular from this study is that the polar Arctic community is particularly sensitive to changing temperature. So as the temperature increases in Arctic, we predict that this plankton interactome will just kind of generally disintegrate because it is particularly sensitive to changing temperature. What we can do with that Arctic community then is subsequently define metrics of what sort of species may be Arctic indicator species, which is shown here in blue. And then for each of these indicator species, we can define a keystone index, which gives a kind of measure of its importance within the whole ecosystem. And we see that typically these indicator species have high keystone indexes, which implies that these organisms are particularly sensitive, particularly important in the ecosystem. And if those species disappear, then that is perhaps going to generate problems for the whole structure of the whole ecosystem. So these are kinds of approaches which were performed using deep learning approaches to sort of get an idea of which organisms are indicator species and their keystone importance in the ecosystems. So those are three examples of what we can do with Tara Ocean's data to explore sensitivity of marine ecosystems to climate change. And I think I will end there in the interest of time, and thank all of the sponsors of the Tara Ocean's expedition and the funders of the Tara Ocean Science and the different laboratories involved in the Tara Ocean's research, which are numerous, too numerous to mention, but they're shown in this slide. So thank you for your attention, and I'll be happy to answer any questions.
Thank you, Dr. Bowler, for this very interesting and informative presentation. We do have some part of plankton in marine biomass. What does it represent in terms of capitation and respiration?
So all of the photosynthetic organisms in the ocean that I mentioned, like cyanobacteria, like diatoms, like dinoflagellates, these collectively, they represent the plants, the trees of the ocean. It's amazing, we don't see them. You know, the ocean is blue, it's not green like on land. But these organisms collectively, in terms of photosynthesis, they generate as much fixed carbon as does all of the photosynthetic trees and plants on land. So they're incredibly important for producing biomass through photosynthesis and through also generating the oxygen that we all need to breathe. So they are as important as the plants and trees on land, and they fuel the food webs in the ocean that go all the way up to the whales, the dolphins, and so on. So without plankton, there would be no other life in the ocean.
Thank you.
It's performing a new expedition called Microbiomes Expedition, trying to explore more about the microbial world in the oceans. So they're performing studies in Antarctica, and then we'll be off the western coast of Africa. And then from 2024, when that mission is finished, there are plans, in fact, to bring Tara back into Indian Ocean and maybe go down towards Kerguelen Islands in Indian Ocean in the south. So there's good chances that Tara will pass by India in a few years' time again. We very much hope so.
Thank you, Dr. Gordon. I think with this question, we'll end the session now. We have to start with our scientific workshops. So thank you once again, Dr. Bola, for taking time out from your schedule to be with us. Thank you very much.
Thank you very much. Pleasure. Thank you.
We shall now move on to the scientific workshops. To attend these workshops, I now request our audience members to go to the lobby area, where you will see 5 different entrances or doors for the workshops. You can attend these workshops according to your interest. These workshops will continue till 5 today, and again tomorrow from 2 to 5. So yeah, you can select the workshop that you are interested in and join those. We will now end the session, and we look forward to seeing you in the workshops. Goodbye.